talk is next generation HCV vaccine antigens, what we know about HCV neutralizing antibodies and responses and how to leverage recent knowledge in vaccine design. Mansun?
Can you hear me?
Yes, thanks,
Yes.
Mansun. We can.
Yeah. Okay. Yeah, thank you so much for the invitation and provide me the opportunity to share my thoughts about this HCV vaccine problem. So to this audience, I don't need to introduce too much about the cause of HCV infection, and over a long period of time, it will induce a lot of inflammation and reinfections of the liver, changing some architect. And one of the key problem really is the HCV, some known as the silence mutation, silence epidemic, and a lot of infections ongoing, don't know. And having a vaccine pretty much can help stop many of these stages of damaging the liver in the infected patients. So one of the thoughts that my title is talk about the next generation HCV vaccine antigens. So we know that there are two major vaccine candidates being tested in humans. One is the viral vectors vaccines to stimulate protective T cell response, talked by Andrea's course yesterday, and then the E1, E2 candidates to elicit neutralizing antibodies, led by Mike Houghton. And so so far we know the current forms we're not quite sure, but probably not protective, or we haven't have the data to show they can be protection. And these two vaccines, I think there's still a possibility to make them to work if we be able to really maximize the immunogenicity and make it elicit very potent immune response. And we did talk about a little bit about the maybe combining these two vaccines or one form or the other, eliciting both T cell and B cell would be a good approach. So my thought is if this vaccine works, it's great. But if not, we really need to think about what to do. And then that's part of the reason I think we need to have a better pipeline for HCV vaccine to increase our chance. And that come to a point that I think by understanding better the immune response being elicited in infections, also being elicited in vaccinations are important, and for the design of next generation HCV vaccine. So my talk is pretty dense, but I try to provide a more overview and a high level way to look into the problems. And in this case, I try to cover 4 different topics. So the first one is about like what's the basal immune response required for protection. So far, the immune correlate is not clearly defined, but because there's not a HCV working HCV vaccine, so we just based on the information we know T cells are important, neutralizing immune response important, and what the problem is we need to have better resolution or details about those immune response. And Andrea yesterday talked about the T cell response, very interesting and in detail. And I'm going to talk a bit more about the antibody response in the past studies. I will also cover a bit about the different topics I will go on in my talk. So the Chiron discovered HCV with other investigators, and they make this full-length E1, E2 antigen. So a few years ago, I was able to get some of these antigens from Phil Domenza. At that time, he was leading the Novartis vaccine effort. And we done some analysis of this vaccine candidate. It come out very nice, like as a major peak. It's about 6 kilo delta when you run a size exclusion. And the protein cell is very clean. You have to see this E2 band and E1 band. And it formed this non-covalent E1, E2 complex. And it just separate by itself with just SDS without reduction. And you can take this protein and look under EM, and you can see it got a kind of lipid micelle with this lob sticking out of the E1, E2 complex. And itself is actually very good in terms of antigenicity. You can pretty much put 3, 4, I think we try up to 5 now, but this one you show you can put 3 different antibodies fragment. You see they all bind to it. And it actually preserves the antigenicity of the E1, E2 antigens very well. So the studies we did, it's very lucky with the help of Sharon Fei and Rajan Koshi. We brought back all this vaccine anti-sera from that study in the phase 1 clinical trial. That at that time 4 doses, test different doses. In parallel, when doing our analysis, we went to immunize, in our case, it's relatively small animal size of four monkeys with this. In this case, we immunized 5 doses of it. And in between, we also collect some sample to isolate monoclonal antibodies with the help of Robert Lanford. And overall, the immune response quite similar. I mean, in terms of the endpoint titer, antibody binding titer, you can see after each booster up to 3, you can see increase of the binding titer for the monkeys, also pretty good. And when you look at neutralization data, which is against the virus, the autologous virus, that the same sequence as the immunizing antigens. So now you see, you can split into the human study, at least there are some patients, some volunteer, they can elicit pretty good prolonged immune response. Some the immune response, the neutralizing antibodies decline rapidly, and some don't really have very high response. Overall, we see about 75% of the vaccinees can elicit neutralizing antibodies against the virus with the same sequence of the antigens. For monkeys, we immunized 4 of them. We see 3 of them achieve production of neutralizing antibodies. One of them kind of transiently, but never achieved the prolonged neutralizing antibody productions. Then we go look into the antibody response a bit more in detail. First, look into how it binds to different HCV antigens, like different genotypes, with a native, long-reduced, and also a delayed E1, E2. You see overall the human samples that neutralize the virus have pretty good binding cause reactivity against different genotypes. Of course, genotype 1 is better binding. And monkeys, the 3 of them neutralize, it binds pretty well. And because it has higher titer, because we immunize animals with more antigens and also 5 times, I think, achieve higher antibody response titer. And if you look at that, it binds to the native E1, E2 a bit better than the reduced E1, E2. So recognize some of this discontinuous epitope. However, when you look at the virus neutralization, it neutralizes autologous virus well. However, but it doesn't cause we neutralize the other genotypes too well. You see some neutralization, but it's nothing impressive. And this is SA13 is a 5A virus, which is known to be very sensitive to neutralization. And the anti-sera can actually neutralize as well as the autologous virus as well. So recently, a number of us grouped together, want to develop better HCV reagents for vaccine studies. This study led by Justin Bailey and Stephen Fang. So we look into a panel of viruses with diverse sequence, and we done a big neutralization study against some of the quite well-known cross-neutralizing antibodies in the field. And we do notice some viruses are more resistant to the neutralization by these antibodies. Some are more sensitive. So kind of roughly divide into 4 tiers, tier 1, 2, 3, 4. And the tier 1 is more sensitive virus to neutralization by this antibody panel. So we took advantage of the human samples, a bit of the leftover. We don't have a lot left. But if you look at that compared to the one I showed you earlier, this human sample, the one we have, it can neutralize the tier 1 virus, which is sensitive virus, pretty well. But again, once you mix with the more resistant virus, you don't see very much neutralization in most of the sample. The same for the monkeys as well. And now we also look into the specificities of the antibodies. Here is a PEP scan assay, look into the antibodies bind to a range, the overlapping peptides across the E1, E2. As expected, you see a lot of reactivity to the XBR1, the hypervariable region 1, and N-terminal of the E1. But overall, they are quite similar between the humans and the monkeys as well. And based on that, we can't really explain the neutralization. These are not neutralizing sera, and this neutralizes the best. Based on PEP scan, you can't tell the difference. But when you look at the more like the discontinuous epitope by competition ELISA, you do see the sample that can block binding of CDH1, which binding to also compete with the AR3 antibodies group. They usually have a better neutralizing activity. And the ones that could not neutralize are the ones that are not making this type of antibodies. And the same for the non-neutralizing sample of the monkey. It doesn't make the CD81 blocking antibodies. So overall, from the human studies, this analysis, we know the E1, E2 itself are immunogenic, and it can elicit neutralizing antibody response against autologous and also sensitive virus. However, it seems to be not very good against more diverse virus, and it's a bit uncertain whether we can push the antigens itself further to produce cross-neutralizing antibodies. Also, when we analyze the human with the monkey antibody response together, we see they're quite similar to each other in terms of neutralizing activity and what virus they can neutralize, and also the epitope they're binding to. So I want to move on to the next one topic is the challenge is to develop vaccine antigens based on the E1, E2. So we did quite a lot of work with these HCV envelope glycoprotein, and here are at least 3 of the main things about when we want to design vaccine antigens based on this E1, E2. And the first thing is that HCV envelope, we know that it's antigenically variable. If you just try to compare with HIV1, at the base amino acid to amino acid level, HCV actually have a higher mutation rate than HIV. Of course, HCV doesn't have as many insertion deletions and guide compared to HIV. However, itself is extremely mutated or variable virus. So this is a problem in vaccine design. And the other part is when one wants to make a HCV antigens for vaccination, itself is actually quite difficult. I just want to show you some cases here. If you try to make the E2 atoll domain by just cloning the sequence and then make a stable cell lines to express this protein. And in our case, we do first purify the supernatant from a stable cell line using antibody feeding column select for folded protein. And after that, you can do a size exclusion and then run the gel here. Here show you 4 different genotypes. And you see they vary quite a lot. Some of those make better monomers, and most of them still have this aggregate of protein, which contain the folded epitopes to be purified alongside with it. And some got pretty poor monomer productions. And these are the covalent aggregates of the protein. And they're always there, and most likely they're in complex with other misfolded protein. So the question really is those contaminants, will that affect in a vaccine when you produce it? On the other hand, when one look into the E2, it's been a long way now. We can do a lot of engineering by truncating some of the regions of the protein and mapping for the binding antibodies. So doing a lot of manipulation in the past, we can delete chunks of the sequence that are flexible, and then we were able to actually make pure enough protein to solve the first structure of the E2 core domains. And here show you. And the problem is when you try to crystallize or solve the structure of the E2 bisomal normally, it doesn't work, because on the protein it has a lot of regions that are extremely flexible, including the we so-called E2 neutralizing phase, which bind the CTH1. If you take the structure, just do an MD molecular dynamic stimulation, you can see this area here is extremely flexible. The gray is the soft structure, and then these regions just keep moving around on the recombinant protein. So when you make the protein for as a vaccine antigen, for immunization, you can expect the host animals or the vaccine is going to see different presentation of the epitopes. And this one we know is for neutralization, but we don't know the other one. So this should be considered in the vaccine design. And indeed, you can even see protein flexibility around the neutralizing phase of E2 by EM. Here just show you the E2 core protein bound by 3 different Fab fragments, AR1B, 1A2, and XC1, which is on one of the bodily neutralizing mechanisms. And you see the angle between this Fab, they are quite variable. Some almost like a person jumping, and then the other one look like a person standing with the 2 legs quite close by. When you start to average this protein image, EM images, the 2D class, you start to see one of the legs disappear. It seems the results just suggest that even the neutralizing epitopes on the E2 core is moving around, and on EM you can just see it when you look at different class. So the question is when you have the vaccine antigen with all these epitopes moving around, would they be be a good antigen, or you may need something more conformation constraint into the epitopes recognized by the body neutralizing antibodies. So for the E2, E12, it has a lot of problems in terms of trying to produce the protein in high yield and good quality. And whether those misfolded proteins come with the purification may cause trouble when you do immunization. And these are the issues we need to deal with in vaccine design. So next one, I want to talk about the neutralizing epitopes on E1, E2. So classically, when I first started, we isolate a panel of human antibodies, and then we map them, and by competition ELISA, during that exercise, we call there 5 non-overlapping antigenic regions on the E1, E2 complex. And AR3 is the one that overlap with the CD81 binding site, the receptor binding site on the protein. And later on, we kind of start to look into it by overlaying the EM data of the recombinant protein with the structure we have, and then also kind of predict where the E1 is going to be present on the E1, E2. So more recently, Rodger Sanders and Andrew Ward, they actually solved the E1, E2 complex by structure by Cryo-EM. So their paper is still under review. It's currently available on the bioarchives. This one, I just want to show you the main thing is like E2 and E1 seems to be forming these elongated structures on here. And the AR4 turnout is still around the E1, E2 interface, but the binding of the AR4 actually only require E2, but you have to fold in a specific conformation for it to bind. If you just express the E2 active domain AR4, it will not bind. And on E2, I mentioned, we know there's a large surface area that are accessible from body neutralizing antibodies, and which combine 2 areas. One is called front layer, the other is called CD81 binding loop, come together to form the discontinuous antigenic surface. And in the middle of it is relatively hydrophobic for the recognition for neutralizing antibodies. Indeed, many groups, including ourselves, the AR3 series of antibodies, and by Justin and by different groups and Stephen, we isolate many antibodies that are recognizing this E2 neutralizing phase. It is just some of the examples. Some bind to this whole surface area, some bind to more selective regions here, and some bind to this little bit of the so-called AS412, the one moving around on the surface. One of the things that most interesting when you look at these antibodies, many of them are actually belongs to the VH1 using the human heavy chain variable domain VH169. Here, just show this group of antibodies. So, and which is pretty interesting. So we start a collaboration with Stephen to look into this VH169 antibodies more carefully by studying their binding and the structures. And when we look at that, with more antibody sequence and structures, one thing you know, as I mentioned, on the E2 neutralizing data is hydrophobic patch in the middle. And in the VH169 germline genes, the CDR H2, the loop 2, usually have a pretty hydrophobic residues embedded in the germlines as well. That for many of these body neutralizing antibodies used to interact with the hydrophobic surface on E2. And usually these antibodies have a longer than average CDR3 loops. And binding most of the time is dominant by heavy chains. Some of the antibodies you only require antibody heavy chain to bind. Light chain is not involved in the binding. And one good thing is these antibodies usually they don't have many somatic hypermutation like the HIV body neutralizing antibodies. So this antibody seems to be achievable when you go vaccinate by usual standard method, and those hypermutation rates should be achievable by vaccination for this VH169 like body neutralizing antibodies for hepatitis C virus. So in the study, we look into here, just show the structure we originally saw with the antibody fat fragment bind to the E1, E2 in this way. And when we solve more structure of Stephen's antibodies, and now we see there's a different way of the antibodies binding to the E2 antigens. And the flipping slightly different from each other. And this is become look like we are looking into two way of bindings of antibodies. However, the way it binds still using quite similar. The heavy light chain, the CDR loops, the way they interact to it, and depending on the CDR, the hydrophobic CDR2 H2 of the VH169 antibodies. However, when we look at the antigen side, the E2 core, we now found that the front layer of the E2 is actually different. One of the group we call the so-called conformation A, we have the front layer recognized in this way, going across the protein and bind to this group of AR3-like antibodies. But when we look at some other antibodies, for example, 212 antibodies, it bind to a conformations a bit different from what we see with the one we saw many times in the past. Now the front layer seems to be somewhere moved like here, away a little bit further in this upway. And in the meantime, because of that, expose some of the residues usually being covered in this conformation in the back layers of the E2. So now it exposed some of the residues. We always thought when you do mapping, when you mutate it, it always lose binding to CDH1. Now it's kind of really explained like this surface area here is kind of moving around, may facilitates the binding of the receptor. And also the back layers of the E2s can be involved in antibody neutralization. So I want to go back to the humans and monkey study I mentioned to you earlier. So at that one of the study in here, the importance I think for me is in the vaccine human study, there wasn't any cells left for analyzing the antibodies at the monoclonal cellular level. So when we do the monkey study, which provide opportunity to look at that more carefully. So in this case, we harvest the cells, we isolate the plasma plus and generate a panel of monkey antibodies. So we clone about 100 of those monkey antibodies from the plasma plus. And from here, you can see it have pretty wide coverage of different VH genes being used to make the monoclonal antibodies. But one thing strike out you can see is the one that have caused neutralizing activity is the VH1.36 class of antibodies. And when you compare the sequence, actually, this is one of the monkey antibody genes closest to the human VH169 germline genes, which was a bit surprising to us. Wow, in monkeys, the monkeys use a human orphan of the VH169 to make cause neutralizing antibodies. When you compare these antibodies, the VH genes together, you can see the human antibodies from here, they all form the bases, the branches. They are almost equal distance to these monkey antibodies when you do the phylogenetic tree. So they're very closely related. However, when you study how these antibodies recognize E2 for neutralization and solving the structure, now we see something interesting. So the one I mentioned to you, like the classical AR3 binding, you have the E2, you have the heavy and light chain of the fat fragment come in this angle, and this mode of binding. And when you look into the how the monkey antibody bind, now you see the antibodies kind of switch with their light chain swap onto the other side to interact with the core domain. Okay. And also one thing interesting to us is, if you look at how all these human antibodies bind, the hydrophobic CDR2 usually come closer, sit more in the middle, interact with the hydrophobic patch of the neutral E2 neutralizing phase in the middle of it. For the monkey antibodies, actually kind of interesting, come on the side. Now the CDR3 actually more occupies in the hydrophobic area for the monkey antibodies. So we, in this study, we found like, okay, monkey use overall antibody genes to produce antibodies bind to the same neutralizing surface, which is interesting. And they also react or cause neutral HCV. However, the way it recognizes antigen surface is very different. So at this point, really can't be certain, is it real? Is it a coincidence? Or actually the functional and the genetics conserved in this case? And this one actually kind of gives some interesting insight. And last month, the group, the Florian Klein group in Germany, they published these studies, which is extremely interesting to us because it can't really come back to the whole thing. It's what we saw in monkeys also happening in humans. So they go to study, they study a number of HCV elite neutralizers and analyze the neutralizing antibodies at the monoclonal level. In the same way, they find a lot of the neutralizing antibodies belong to VH169. And when they look into more carefully of those antibodies, Here just show some of the examples, like the one they isolate from patients compared to those also isolated from patients by the single B cell method or by recombinant methods. And they all have this cross-neutralizing activity to it. The most striking part is now the antibodies, the way it binds to it, actually more like the monkey's antibodies than what we classically see in humans. And you can see in here, the way it binds, it recognizes in this angle, and then the footprint of how the antibodies bind is almost like the monkeys. And of course, the most strikingly, you see, I explained to you how CDR2 bind with these classical antibodies. It comes in around this, try to interact with the center of the E2 neutralizing phase. And now you look at the new antibodies isolated by the client group, you see the way of the approach of the antibodies, almost like the monkeys. Like the CDR2 is on the top instead of on the bottom. And you can even kind of tell where's the light chain here. In this case, the light chain is on this side, and you can see it classically because of the switch of the antibodies binding mode. It comes from the other angle. So it's really kind of interesting. We found from monkey first a new way of VH169 antibodies neutralize HCV, and then later on we found in humans as well. So because of all these information all come together, there's really a so-called multi-donor class of body neutralizing antibody response when people see the HCV E2 antigens, and some people call it public neutralizing antibodies response. And even more interesting seems to be this class of antibodies that conserve in humans, also in non-human primates. So to summarize a bit about the cross-neutralizing epitope of HCV for vaccine design, we know the E2 neutralizing phase is quite immunogenic. Many antibodies been isolated to it. I mentioned the AL4. Now the structure is out. We can think a bit more how to design vaccine based on that. And there's a multi-donor class as a neutralizing antibody response. And seems to be the non-human primate, the macaque model will be very useful to study this type of neutralizing antibody response. Okay, to finish with my talk, I just want to quickly talk about like some of the strategy with the structural information, with this binding mode, with this multi-donor class of antibodies. One of the key issues, really, we need to think about how to stabilize and better present the neutralizing epitopes. And one thing we didn't talk a lot about, but it's quite essential in terms of vaccine design. You really need to make antigens have a very, very, very favorable properties, good antigenicity, good yield, purity, and stable for vaccination, and easy to produce. And some of the approved human adjuvants, we already tested the CPG. It also worked with E2. We don't know about the LNP that combined with the mRNA vaccine, which now known is also very immunogenic. So the way to improve the immunogenicity, one of the approach we take is try to first optimize our antigens and then put onto these self-assembling protein nanoparticles championed by my colleagues Jiangsu and Scripps. And the idea pretty much, this is a one single protein. It will express in high yield and can be produced at the GMP level. It will cell assembly, relatively easy to produce and form nice nanoparticles. And he has some data to show this nanoparticle are more stable and retain into the lymph node for a while, and then much longer than just the single protein. And it can elicit germinal center B cell and TFH cells much faster than these monomeric protein in comparison for the protein displaced on the nanoparticles. So we know that we can displace E2 on some of these nanoparticles. And now we kind of see continuous optimizing the protein, also see whether we put E1, E2 on top of this nanoparticle and to produce it. So I want to conclude my talk, and we have teams at Scripps Research also with Chris Chen in the Southwest National Primary Research Center to push this HCV vaccine effort using the approach quite different from the others existing one. And I also want to thank for many people in the field and help our efforts and being an extremely good collaborator. Also want to acknowledge the funding from NIH, NIH to support our work. Yeah, thank you.